Good morning, friends. Today we will discuss about immunoprecipitation reaction. Before that, I request all of my friends, please subscribe my YouTube channel and also watch, like and share my videos with your friends. Antigen-antibody interaction is highly specific. It is similar to the interaction between enzyme and substrate. Antigen-antibody binding involves weak, reversible and non-covalent interactions such as van der Waals force, electrostatic force, hydrogen bonds and hydrophobic association. The smallest unit of an antigen that binds antibody is called antigenic determinant antigenic determinant or epitope The corresponding area on an antibody that binds with epitope is called paratope. The number of epitopes on the surface of an antigen is its valency. So valency is the number of epitopes present on the surface of an antigen. It determines the number of antibody molecules that can combine with an antigen at one time. If one epitope is present, then the antigen is monovalent. Most antigens, however, contain more than one copy of the same epitope and are therefore polyvalent. Immunoprecipitation reaction is the interaction between soluble antigens and soluble antibodies that result in the formation of large insoluble antigen-antibody complex. The formation of antigen-antibody lattice depends on the valency of both antibody and antigen. The antibody must be bivalent for immunoprecipitation to occur. Similarly, antigens must be bivalent or polyvalent. When the antigen is polyvalent, then it binds with multiple antibodies. Due to such interaction between antigen and antibody, a cross-linked antigen-antibody complex is formed which becomes so large that it precipitates out of the solution. Immunoprecipitation reaction can be performed in solution or in gel. First one is immunoprecipitation reaction in solution. Immunoprecipitation occurs due to the formation of large insoluble antigen-antibody complex. Precipitation occurs maximally only when there are optimal proportions of antibody and the antigen. Maximum precipitation takes place within a narrow concentration range known as the zone of equivalence. This figure explains the principle of immunoprecipitation. These Y-shaped structures are antibodies and these blue circles are antigen. In the zone of equivalence, antigen and antibody are in equal proportion. In this zone, due to maximum cross-linking, antigen-antibody complexes are formed that are sufficiently large. These large complexes become insoluble and precipitate out of the solution. In this figure, you can see that these antibody molecules are cross-linked by antigen. This interaction between antibody and antigen results in the formation of large insoluble complexes. As a result, visible precipitation takes place in the zone of equivalence. On one side of the zone of equivalence, there is antibody excess. In this zone, the concentration of antibody is greater than the concentration of antigen. In this zone, cross-linking between antibody and antigen is minimum, and because of this, small soluble antigen-antibody complexes are formed. Because of this, in the zone of antibody excess, precipitation is inhibited. On other side of the zone of equivalence, there is another zone in which antigens are in excess. In this zone, the concentration of antigen is greater than the concentration of antibody. Like the zone of antibody excess, in this zone also, antigen and antibody are not in equal proportion. This results in the formation of small soluble antigen-antibody complexes. Because of this, in the zone of antigen excess, precipitation is inhibited. When increasing amount of antigen is added to a series of test tubes containing a constant amount of antibody, variable amount of precipitate is formed. When the amount of precipitate is plotted against different antigen concentration at which it is formed, a curve is obtained. This curve is known as the precipitation curve. The middle segment of the curve represents the equivalent zone in which precipitation is maximum. On both sides, in the zone of antibody excess and also in the zone of antigen excess, visible precipitate is not formed. In this graph, you can observe that when we increase the antigen concentration, rate of precipitation also increases gradually. At the equivalence zone, precipitation rate becomes maximum. After the equivalence zone, as the antigen concentration is increased further, precipitation rate decreases. Immunoprecipitation reaction is used to remove antigen from a solution. Second one is immunoprecipitation reaction in gel. Immunoprecipitation performed in a gel is known as immunodiffusion reaction. Immunodiffusion reaction. There are two types of immunodiffusions that are performed to determine the relative concentration of antigen, antibody, as well as the identity of antigen. These two techniques are radial immunodiffusion and double immunodiffusion Radial immunodiffusion is a simple quantitative assay. It is also known as Mancini method. It is also known as Mancini method. In case of radial immunodiffusion, the sample containing antigen is placed in a well. The antigen is then allowed to diffuse radially outward into the gel containing antibody. This is the gel in which antibody is uniformly distributed. So antigens present in the sample move radially outward and in the zone of equivalence. Because antigen and antibody are in equal proportion, maximum precipitation takes place and a precipitin ring is formed around the well. The diameter of the precipitation ring is proportional to the concentration of antigen present in the sample. In order to determine the concentration of antigen in the sample, different amount of the standard antigen and also the sample are subjected to radial immunodiffusion in one gel. After performing the procedure, the diameter of the precipitation ring were measured. A standard calibration curve is then plotted by taking the diameter of precipitin rings on the x-axis and the log of concentration of antigen on the y-axis. The concentration of antigen in the sample is determined from this calibration curve. Radial immunodiffusion is used to determine the relative concentration of antigen in a sample. Double immunodiffusion is also known as octolony double immunodiffusion. In this technique, both antibody and antigen are allowed to diffuse toward each other. As a result, distinct precipitation lines are formed in the zone of equivalence. Double immunodiffusion is used for the detection, identification and quantification of antibody and antigen. The relationship between two antigens can also be analyzed by this technique. Distinct precipitation lines are formed against the same antibody depending whether the two antigens are identical, non-identical or partially identical. This figure illustrates as how double immunodiffusion can be used to analyze relationship between two antigens. When two antigens are identical, then a precipitation arc is formed between antibody and antigen at the zone of equivalence. But in case two antigens are not identical, two crossed precipitation lines are formed against the same antibody. But for antigens that are partially identical, a precipitation line is formed for the common epitope, but a curved spore for the unique epitope. Friends, this is all about different types of immunoprecipitation reactions. For more updates, please subscribe my YouTube channel, Sam Biology Class. Have a nice day.